biosynthesis of insulin involves a precursor molecule that is pre-proinsulin. Pre-proinsulin in presence of proteolytic enzyme gets converted into proinsulin in the rough endoplasmic reticulum. The proinsulin in presence of proteolytic enzymes such as proteases gets converted into insulin. The proinsulin consists of two polypeptide chains, that is A and B chains, with a connecting peptide, that is C peptide, consisting of amino acids. In presence of enzymes such as proteases, the C peptide or the connecting peptide is removed and the proinsulin gets converted to insulin. So after insulin is synthesized, it is stored in the granules of the pancreatic beta cells and is released into the systemic circulation. So what causes the release of insulin into the systemic circulation? Glucose at a concentration greater than 70 mg per deciliter stimulates insulin secretion. So let's see how does glucose stimulate the secretion of insulin. As we can see in this figure, GLUT2 transporters are expressed in the beta cell membrane, and this facilitates the entry of glucose into the pancreatic beta cells. Once the glucose is inside the pancreatic beta cells, it undergoes phosphorylation in presence of glucokinase enzyme and forms glucose 6-phosphate. The glucose 6-phosphate further undergoes oxidation to generate ATP. ATP acts on the ATP-sensitive potassium channels, causing inhibition of the potassium channels and causing depolarization of the beta cell membrane. Because of depolarization of the beta cell membrane, the calcium channels open up, allowing the entry of calcium ions. As we know that calcium stimulates secretion, so calcium causes docking or fusion of the vesicles with the beta cell membrane, and this causes the vesicles to secrete insulin into the systemic circulation. So insulin is released into the systemic circulation and is available for action. So now that insulin is available in the systemic circulation for action, insulin facilitates the uptake of glucose through GLUT4 transporters that are expressed in the skeletal muscle and adipose tissue. So GLUT4 transporters are predominantly expressed in the skeletal muscle and adipose tissue, and these transporters facilitate the glucose utilization by the skeletal muscle and adipose tissue. So how does insulin act? Insulin acts on the insulin receptor, which is an enzyme-linked receptor. The insulin receptor has two alpha chains and two beta chains linked together by disulfide bonds. The two alpha chains has a ligand binding domain to which insulin binds, and the beta chains are attached to tyrosine kinase enzyme residue. So the binding of insulin causes activation of tyrosine kinase enzyme, which causes autophosphorylation of the beta chains and phosphorylation of the tyrosine residues of the insulin receptor substrate protein. So this causes activation of signaling pathway, that is phosphoinositol 3 kinase pathway, and a series of events of phosphorylation and dephosphorylation occurs. Because of the activation of the signaling pathway, there is transcription, protein synthesis, and the GLUT4 transporters are synthesized and translocated to be expressed in the cell membrane of the skeletal muscle and adipose tissue. So because of the expression of GLUT4 transporters on the skeletal muscle and adipose tissue, there is glucose utilization by the skeletal muscle and adipose tissue for action. So this is how insulin facilitates glucose utilization by the skeletal muscle and adipose tissue.